Uh, Good afternoon or good morning, everyone. Welcome to our Clinical Pharmacology Training Network webinar sponsored by the Imprint Hub. Today, our speaker is Dr. Bernd Meibohm, who will be speaking on pharmacokinetics and pharmacodynamics of therapeutic proteins in pediatrics. Um, Dr. Meibohm is a distinguished professor of pharmaceutical sciences and associate dean for research and graduate programs at the College of Pharmacy at the University of Tennessee Health Science Center in Memphis, Tennessee. He also serves as chair for the University of Tennessee Department of Pharmaceutical Sciences and holds the Harriet S. Van Fleet Endowed Professorship in Pharmaceutics. Dr. Meibohm is well recognized for his scientific career, including interest in bacterial and viral infectious diseases, pediatric pharmacology, and application of pharmacometric modeling and simulation techniques, with a specific focus on therapeutic proteins. He is also involved in leadership positions with the American Association of Pharmaceutical Sciences, the American College of Clinical Pharmacology, and the International Society of Pharmacometrics. We thank Dr. Meibohm for joining us today, and Bernd, I will turn it over to you for the presentation.
Well, thank you very much for the introduction, Sarah. Good morning, good afternoon, everyone. So let me share my slides with you, and then we go right away into our presentation. So I will talk a little bit, as Sarah said, about pharmacokinetics and pharmacodynamics of therapeutic proteins in the arena of pediatric pharmacotherapy. Just my disclosures, I serve as consultant for a variety of pharmaceutical companies, and the learning objectives. So we'll focus on understanding the mechanistic basis for drug disposition of therapeutic proteins and how they differ from small molecule drugs. I would like you to appreciate the impact and the challenges of drug-target interactions on drug disposition. We'll explain the impact of immunogenicity on the PK and PD of therapeutic proteins, and ultimately identify factors of the PK and PD of therapeutic proteins that are modulated by childhood development. When you think about therapeutic proteins, then shown here is the last 30 years of drug approvals by the US Food and Drug Administration. And you see differentiated in blue the new molecular entities, the traditional small molecule drugs, and in red the biologic license applications. And the biologic license applications include therapeutic proteins, but they also include vaccines, for example, or blood products. But what you clearly can see that, especially over the last 10 to 15 years, approximately a third to a quarter of all drug approvals have been biologic license applications. That means by now there is a large fraction of our pharmacopoeia that ultimately consists of therapeutic proteins that are regularly used in a variety of different indications. More importantly, from the drug development standpoint, but also from the healthcare system standpoint, is that the top 10 medications in sales by 2024 that are shown here, 7 of those 10 medications are therapeutic peptides or proteins. So therapeutic peptides and proteins are also a major moneymaker for the pharmaceutical industry. They oftentimes address unmet medical needs in many different indications, but they also are a major cost factor in the healthcare system for prescribers, for hospitals, and for insurance companies. So clearly there's a lot of focus on these kind of medications, and this is not only limited, of course, to the adult populations, but has also increased more and more in the pediatric space. So when we compare classic small molecule drugs to therapeutic proteins, and small molecule drugs are defined chemically by a structure and by their purity. So we know exactly what we have in front of us. And if I buy today a bottle of a small molecule drug and compare this with what I bought last year, then it's exactly the same. So it's chemically synthesized. Examples are your fentanyl or ibuprofen, and they are identical from batch to batch. When we look at therapeutic proteins, the major difference is that therapeutic proteins are not chemically synthesized, but they are produced in living organisms, usually in genetically modified cells, either bacterial cells or mammalian cells. Most complex therapeutic proteins nowadays are produced in Chinese ovarian hamster cells, so in mammalian cells. And since they are produced by a living organism, they are not defined by their structure or purity, but they are defined by the production process. And this production process needs to be extremely carefully controlled so that these living organisms that have been modified to produce or overproduce a certain therapeutic protein of interest, that these living organisms, these cells, are actually are in an environment where they actually produce the kind of therapeutic protein you would like to have. So that means you have to have very defined CMC criteria in the production process, but you also have a large number of so-called critical quality attributes, so measures, physiologic, functional measures of this drug product that ultimately altogether characterize a drug product. For a large therapeutic protein, like a monoclonal antibody, there are approximately 80 to 120 different critical quality measures that have been carefully defined and that need to be maintained within a certain predefined range to ultimately ensure that the drug product that is produced, the therapeutic protein that is produced, has consistent efficacy and safety when used in human patient populations. So that means each drug product is unique, even though it may be very similar to something that's already on the market, and the whole concept of biosimilarity is based on that you cannot produce a drug compound from an innovator drug, for example, that's identical to it, but you can produce something that's highly similar based on these defined production process, and that highly similar product will then ensure that you have similar efficacy and safety when used compared to an innovator drug. That also means that batches are not identical, but they have, again, based on this carefully controlled production environment, they are highly similar. And that is what needs to be established within the production process of a therapeutic protein. Just to remember what proteins in general are, proteins are linear polymers of amino acids. So amino acids, 20 essential amino acids, are the building blocks for our proteins that we have in the body, and dependent on how these amino acids are arranged, they are basically fused together by peptide bonds, as shown here, and then you end up basically with this long linear chain of amino acids, which is called the primary structure of the protein. Now, a protein is not only defined by its primary structure, but also by its secondary, tertiary, and quaternary structure, which are basically the three-dimensional arrangement of this amino acid chain, this linear polymer of amino acids. And the reason for that is that the linear polymer is energetically unstable in a aqueous environment. So it starts to fold up. Those areas that are more hydrophobic try to get in outside of the vasculars of the aqueous environment. So basically form the inner part of the three-dimensional structures, and those that are more hydrophilic become the outside structure. So you basically get a very defined folding, and this folding is crucial for maintaining the function and the activity of a therapeutic protein. Now, in contrast to the peptide bonds for the primary structure, the secondary, tertiary, and quaternary structures are supported by relatively weak interactions. So not atomic bonds, but by van der Waals interactions, by dipole-dipolar interactions, by hydrogen bonding. And by that, you have a three-dimensional structure that's crucial for activity and function of the therapeutic protein, but it's also relatively labile. And that means that you cannot put too much stress on a therapeutic protein, stress with regard to temperature, for example, increased temperature, but also with regard to shear stress. For example, if you try to squeeze a protein solution through a very small needle at high speed. Those stress conditions would ultimately break up the three-dimensional structure, and by that, the protein would lose its function. So that also means that many therapeutic proteins have to be stored, for example, by refrigeration to minimize the stress. You cannot freeze them. That would also destroy the three-dimensional structure, but at the same time, you cannot store it at room temperature or in an increased temperature environment if you are, for example, in a tropical region. Now, if we talk about therapeutic proteins, then we also have to appreciate that we have substantial structural diversity. So shown here as an example is a very small therapeutic protein, insulin, which has a molecular weight of 5.8 kilodalton and consists of 51 amino acids, compared to albumin, a structural protein in the body, rarely used for therapeutic purpose by itself, but oftentimes fused together in the so-called fusion protein with other components, and by that also used as in therapeutic scenarios. So albumin has a molecular weight of 66.5 kilodalton and consists of 583 amino acids. And then a stereotypical monoclonal antibody, so in immunoglobulin G molecule, which has a molecular weight of 150 kilodalton and consists of 1,300 amino acids. So if you compare these three, the small, the insulin versus the monoclonal antibody, then you can imagine that the differences in size, in charge, and in structure ultimately are major drivers for the behavior of these molecules when they move throughout the body, through the vascular system, through extravascular systems, and ultimately interact with target structures. So protein structure is the major driver for their behavior in the body. And if we talk about proteins, we always need to remember what kind of protein we talk about with regard to structure and size. So the major classes of therapeutic proteins that are clinically used are, of course, monoclonal antibodies and antibody constructs or derivatives. So monoclonal antibodies like bevacizumab in cancer or infliximab in rheumatoid arthritis. Then we have modified monoclonal antibodies where certain small molecule components have been added to. These are the antibody drug conjugates, brentuximab vedotin for the treatment of lymphoma as an example. And then we have more newer constructs that are based on the monoclonal antibody backbone, but have been substantially structurally modified. Two examples are listed here. Ranibizumab is a FAB fragment that's injected in the eye for the treatment of age-related macular degeneration, and blinatumomab, which is a so-called bispecific antibody fragment that's used to treat acute lymphoblastic leukemia. The second major class are hormones and growth factors, either identical to endogenous hormones or growth factors or modified to enhance certain functions of these growth factors. Insulin is, of course, a classic example for the treatment of type 1 diabetes, filgrastim as a neutrophil growth factor for the treatment of neutropenia, and beta interferon for the treatment of multiple sclerosis. And then the third large group is the so-called enzyme replacement therapy, used usually in rare diseases where you have hereditary conditions in patients where certain endogenous enzymes are either non-functional or only expressed in a reduced level, and by that you have to receive lifelong replacement therapy. Agalsidase beta in Fabry disease is an example for a classic enzyme replacement therapy. So Luckily, the central paradigm of clinical pharmacology, as we know it from small molecule drugs, is also more or less equally applicable to therapeutic proteins. So basically, we give a dose or dosing regimen to a patient that results in time courses of concentrations in different organs and tissues, and this relationship between the dosing regimen and these concentration time courses are characterized by pharmacokinetics. And then these time courses of concentrations are the driver for the desired or the undesired effects in the patient, so either the efficacy or the toxicity, and the quantitative relationship between these time courses of concentration in different organs and tissues and the observed desired or undesired effects are characterized by pharmacodynamics. So in the following, I would like to focus on these 5 different aspects of drug disposition of therapeutic proteins. So I would like to start with absorption processes. Now, as most of you are aware, therapeutic proteins are usually given by intravenous administration, either at infusion or injection. And the reason for that is that they lack oral bioavailability. And there are 2 reasons for that. One is that the gastrointestinal tract is the most efficient metabolism site for proteins, for, of course, proteins that are taken up for nutritional purposes. So you have proteases that break down these proteins, and they do not differentiate between therapeutically administered proteins versus nutritional proteins. But the more important aspect is not the gastrointestinal activity, because you can, it has been shown that if you give the protease inhibitors and you block gastrointestinal protease activity, you still do not get substantial oral bioavailability if you give a therapeutic protein by that route. So the major obstacle is actually the low permeability through the gastrointestinal mucosa. So ultimately, the size, the large size, and the charge of therapeutic proteins is ultimately the major obstacle for their uptake into the systemic circulation if you would give them by the oral route. So that it means we have to give intravenous administration, or we have to give another parenteral administration, which is either intramuscular or subcutaneous administration. And that, of course, is very popular for self-administration. So some of these devices that are used for subcutaneous administration are shown here on the right side. On the top, a adalimumab pre-filled syringes for subcutaneous administration, or a pen device on the upper right side. And then on the bottom, you see a growth hormone pen for multiple use, where you actually have a dial and the patient can select the dose to be administered and then press the button to initiate the subcutaneous administration. Alternative administration routes have, over the last decades, been explored. One that is used for many years is intranasal administration. For example, desmopressin is a small peptide that is administered that way, and it works very reliably and very well. But intranasal administration has major limitations with regard to the quantities you can deliver to a patient. Pulmonary administration, so inhalation, has been explored. There was at some point, and there currently is, a insulin drug product on the market that can be inhaled, but it still has only a niche indication, so it's only used in a very limited patient population. So really, a parenteral administration, either IV administration by infusion or injection, or subcutaneous administration, are the major routes of administration. Now, if we give subcutaneous administration, then this slide is here to remind you of the tissue physiology in the subcutaneous space. We obviously have capillary beds there, but we also have a lymphatic bed. And the lymphatic system plays a major role in the drug disposition as well as the absorption of therapeutic proteins. And what you can see here is if a little graphic where you have a dependency of the lymph recovery of a subcutaneously administered dose of a therapeutic protein versus molecular weight. So on the left side here, 5-fluoro-2-deoxyuridine is a small molecule. Inulin, still a small molecule, larger molecular weight of sugar, basically. Then a small peptide and a slightly larger one with cytochrome C and interferon alpha 2A. And you see with increasing size, after subcutaneous administration, you have an increasing percentage of lymph recovery. And that actually could be shown in other studies that you can actually extend this line. And that means that you have large molecular weight proteins like a monoclonal antibody with 150 kilodalton. You basically have, after subcutaneous administration, nearly complete recovery in the lymphatic system. So after subcutaneous administration, the larger the molecular weight of the drug, the higher the percentage that's absorbed into the lymphatic system, and not in blood capillaries. And for a large therapeutic protein, this is approximately 100%. So what are the consequences of this? Well, one consequence is that you get a very slow and protracted absorption process. And that is related to the much slower flow rate of fluid in the lymphatic system compared to the vascular system. The flow rate in the lymphatic vessels is approximately 0.2% of the flow rate that you have in the vascular system. So approximately 500 times slower. And that really means that whatever is taken up into the lymphatic system has a long residence time in that lymphatic system before it actually reaches the venous bloodstream through the thoracic duct and the subclavian vein. In addition to that, lymphatic vessels obviously drain into lymph nodes, and lymph nodes are rich in phagocytic cells. So that means this long residence time in the lymphatic system provides a lot of opportunity to for phagocytic cells in lymph nodes, but also for endothelial signs that line these blood vessels to take up the therapeutic protein and ultimately degrade it. And that means that after subcutaneous administration, you have not only a substantially protracted absorption process, but you also have a substantially reduced bioavailability. So for most therapeutic proteins, the bioavailability after subcutaneous administration is only 40 to 65% of what you achieve after intravenous administration. So you have to give much larger doses subcutaneously to ultimately achieve the same kind of drug exposures in the systemic circulation compared to an IV administration. Now, what's different in pediatric patients? Well, the route of administration for older pediatric patients is similarly preferred subcutaneous administration compared to IV or IM administration. However, for very young pediatric patients, as you're well aware, intramuscular administration and IV are often preferred, and of course intramuscular due to the ease of injection into the vastus lateralis of the thigh compared to a subcutaneous administration. Now, after subcutaneous or EM administration in young children, an increased rate of absorption has been observed compared to adults. That can be explained with increased extracellular fluid volume in young pediatric patients, as well as with higher perfusion rates, not only in the lymphatic system, but also in the vascular system compared to older children and adults. And that is, for example, reflected in the administration of palivizumab, an older monoclonal antibody for the treatment of respiratory syncytial virus, which has shown to have a 3 times faster absorption rate in children compared to adults. Interestingly, there's no difference in the extent of the absorption. So this is potentially related to the increased extent of absorption is counterbalanced by an increased endosomal protein turnover in children, as we will talk about in a few minutes. So now let's move from absorption to distribution and tissue penetration processes. In contrast to small molecule drugs, therapeutic proteins are distributed not by diffusion, but by convective extravasation. And convective extravasation follows the fluid flux from the vascular space into the interstitial space and then drainage through the lymphatic system. So there's a constant flux of fluid, of plasma water, basically, through paracellular pores between um, uh, endothelial cells that line these blood vessels into the interstitial space. And then in the interstitial space, you have, again, convective transport into the lymphatic system. Now, this transport is facilitated by differences in hydrostatic as well as osmotic pressure. And it's also characterized by the fact that you have a much larger number of pores and much larger pores between endothelial cells lining the lymphatic system compared to the vascular system. And that means that it's much easier to transfer from the vascular system. Sorry, it's much easier to transfer from the interstitial space to the lymphatic system compared to the transfer from the vascular system to the interstitial space. So that means that you have much faster transfer clearance into the lymphatic system from the interstitial space compared to the extravasation clearance from the vascular space. That means that the concentrations in this interstitial space always stay much lower than the concentrations in the vascular space. So the differences in convective uptake into tissues and convective elimination via the lymphatic drainage are the major driver for distribution of large therapeutic proteins. And the concentrations in the interstitial space are substantially lower than in plasma. If you look at endogenous immune globulin G concentrations, they are approximately tenfold lower in many tissues compared to plasma. Of course, you have tissues with where the endothelial cells are characterized by more and larger pores, so more leaky capillaries, basically. And in those, of course, these concentration differences are less pronounced. So distribution processes are largely driven by molecular size and charge, and that means for large therapeutic proteins like monoclonal antibodies that have a high molecular weight, they are largely confined to the vascular space and to a much lesser degree entering the interstitial space. Usually you have, after intravenous administration, a bi-exponential concentration time profile that ultimately is characterized by an initial distribution volume that's basically in the range of the plasma water, 2 to 3 liters, and the overall volume of distribution is not much larger than 5 to 20 liters. And that's very consistent across most therapeutic proteins. Now, if you look at pediatric patients, it's well known that there are differences in tissue water between newborns and young infants compared to older children and adults. The fraction of total body volume available for distribution is higher in children for hydrophilic macromolecules such as monoclonal antibodies, and this is simply driven by this graphic here, or by the fact that's shown in the graphic here, that you have a much higher percentage of body weight that is water in young pediatric patients compared to adults, and also that the distribution between intracellular and extracellular fluid is driven towards the extracellular fluid in young pediatric patients. So more extracellular fluid available, by that you have a higher volume of distribution for hydrophilic macromolecules such as monoclonal antibodies. In addition, the capillaries in infants, you usually have larger capillary beds and thus larger capillary surface area per volume of tissue compared to adults. And you have a larger proportion of so-called leaky organs and tissues like liver, kidneys, and spleen relative to body size. And all this together basically is expected to result in faster extravasation and a concentration difference between vascular and extravascular drug concentrations that are lower in neonates and infants compared to older children and adults. So now let's focus on elimination processes as major drivers for drug exposure. The most important elimination process for therapeutic proteins is the so-called non-specific proteolytic clearance. And non-specific proteolytic clearance uses the same catabolic pathways as are used in the body for endogenous or dietetic proteins, which are ultimately broken up into amino acids, and these amino acids are reutilized in the endogenous pool to synthesize new structural or functional proteins. So your therapeutic protein ultimately never leaves the body. It's just broken up into its building blocks to the amino acids, and those are reutilized endogenously. Now, where does proteolytic degradation occur? It occurs primarily in endothelial cells. And endothelial cells, of course, line blood vessels as well as lymphatic vessels and are a major contributor due to endocytosis. And this endocytosis process is relatively slow and inefficient. However, endothelial cells altogether create a large surface area, approximately more than 1,000 square meter in adult human. So all these cells together that face ultimately the vascular space. And by that, even though endocytosis is inefficient and slow, it still is a major contributor to uptake of therapeutic proteins into these endothelial cells and then subsequent intracellular lysosomal degradation. Now, the major organs that are then contributors for non-specific proteolytic degradation through endothelial cells are those organs and tissues that have the largest capillary beds, and that are the skin, the intestine, and the muscle. So skin, intestine, and muscle are the major contributor to protein degradation. In addition, the reticuloendothelial system, so mononuclear phagocytic cells like macrophages, monocytes, Kupffer cells, Langerhans cells, depending on where these phagocytic cells are located in which organ and tissue, all contribute also to the breakup and endogenous degradation of therapeutic proteins. So the reticuloendothelial system together with endothelial cells are the major contributors. Now, other organs may be minor sites of degradation, like liver, kidney, spleen, and lungs, but for specific therapeutic proteins, for one or the other, one of these organs may also be a major contributor. Now, the major or the rate limiting step in this proteolytic degradation is the intracellular uptake. And I already mentioned the endocytosis through as a relatively unspecific process, especially if it's mediated through pinocytosis. And pinocytosis is a fluid phase endocytosis where a small droplet of extracellular fluid is basically taken up in a vessel in an endothelial cell. So this is relatively slow and relatively inefficient, but due to the large surface area of endothelial cells, still makes a major contribution. What are much more efficient and faster uptake processes is endocytosis that's mediated through membrane standing receptors. And there are two kind of endocytosis processes that can be differentiated. The first one is through unspecific promiscuous membrane receptors, like the LDL receptor or sugar recognizing receptors, so receptors that can recognize structural features in a variety of different therapeutic proteins, and by that can interact with many different therapeutic proteins that have this specific structural feature, like a end-standing sugar chain, for example, attached to them. More specific is then the so-called target median endocytosis, and that's basically the binding of the therapeutic protein to again, a membrane standing receptor, but this receptor is now the target receptor for that specific protein. For example, if you have a monoclonal antibody that targets a specific membrane standing receptor like HER2, then this interaction with HER2 would be lead to binding to this receptor and then internalization of the therapeutic protein bound to the receptor into the cell, again, into an endosome. This endosome is processed to become a lysosome, and you get lysosomal degradation of the internalized therapeutic protein. Now, target-mediated drug disposition is a feature that many therapeutic proteins show, and it is basically the interaction, as I just showed you, with its target receptor. And shown here is in this little cartoon a pharmacokinetic model, a two-compartment model for your therapeutic protein of interest, and this therapeutic protein of interest then can bind to your target receptor, and that target receptor follows, of course, also endogenous synthesis and degradation, turnover kinetics, basically. So ultimately, you form a drug receptor complex, and that drug receptor complex elicits its effect. And this drug receptor complex can be formed, it can be broken up again, so it has K on and K off kinetics, very similar to what we are used to for small molecule drug. The major difference now is for therapeutic proteins that this drug receptor complex can be internalized in the cell. It's a membrane standing receptor, and then basically can be degraded. And it would lead to the degradation of the receptor as well as the therapeutic protein. So ultimately, it is an additional elimination process. So we do not have this separation between pharmacokinetic drug disposition and pharmacodynamics anymore, but the pharmacodynamics in this case, the interaction of the therapeutic protein with its target receptor to form the drug receptor complex that elicits the effect, is also a pharmacokinetic process by constituting an additional endogenous elimination process. Now, since this is a receptor-mediated process, you can easily give drug amounts that start to saturate the available receptors within the body. And what you then get is so-called nonlinear pharmacokinetic behavior. And that's very frequently observed for therapeutic proteins, nonlinear pharmacokinetic behavior. Now, the kidneys, I mentioned, is usually only a minor contributor to elimination for therapeutic proteins. And of course, the kidneys do not excrete proteins. If proteins are detected in your urine, obviously this would be a pathophysiologic condition, usually related to some damage in the kidneys. So therapeutic proteins are not excreted into the urine in unchanged form. However, if they are small enough to undergo glomerular filtration, they can still be metabolized in the kidney. So they will be filtered, they will enter the primary urine in the proximal tubulus, and that usually occurs when their size is smaller than 60 kilodalton. Of course, there's not a molecular balance in the kidneys, so what is really the driver is the effective molecular radius that allows to get through the filtration membrane of the glomerulus. But ultimately, as a rule of thumb, the size of approximately 60 kilodalton is the rate limiting factor. So that means for therapeutic proteins that are much smaller than 60 kilodalton, for example, 5 kilodalton, they can be filtered very efficiently with a filtration rate that reaches the glomerular filtration rate. Once they get larger than 30 kilodalton, the filtration rate falls off sharply. And there's also some effect of charge selectivity in addition to the size selectivity, where you get a better filtration with cationic compared to anionic molecules. So what does this mean then for patients that have renal impairment? Well, that means that if you have a therapeutic protein with a molecular weight of less than 60 kilodalton, you need to be concerned about renal impairment. And that, for example, could be shown for recombinant human interleukin 18, which has interleukin 10, sorry, which has a molecular weight of 18 kilodalton, so much smaller than the 60 kilodalton cutoff. So that means it is filtered in the kidneys and then undergoes metabolism in proximal tubular cells. So it's taken up from the primary urine by endocytosis and is then processed in these proximal tubular cells. And that means with decreasing renal function, measured here as creatinine clearance, so the lower creatinine clearance is decreased renal function, you get an increased systemic exposure, a longer half-life for the same dose of recombinant human interleukin 10. So clearly, structure drives the behavior in the body, structure and size, as I tried to convey earlier. If you have a molecular therapeutic protein that is smaller than 60 kilodalton, it can undergo glomerular filtration and subsequent renal metabolism. By that, patients with renal impairment need to be carefully dosed because the renal impairment will reduce the clearance for these drugs and by that increase the systemic exposure as shown here for interleukin-10. What does this mean then with regard to therapeutic proteins that have a molecular weight of larger than 60 kilodalton, for example, a monoclonal antibody? Well, that's shown in this example here for elotuzumab, where a study was performed in patients with end-stage renal disease and severe renal impairment with a creatinine clearance below 30 milliliters per minute compared to normal renal function individuals. And what you can see is that the concentration time profiles, the averages here are overlapping. So there is no difference, no impact of renal impairment on antibody disposition because antibodies have a molecular weight that is much larger than this cutoff of 60 kilodalton. Now, what happens then if we talk about renal impairment? We also need to talk about hepatic impairment. What happens with hepatic impairment? As I mentioned, there is no impact for most therapeutic proteins since the liver is only the major site of metabolism for very few therapeutic proteins, and most of the elimination is facilitated throughout the body by endothelial cells that line blood vessels, as well as by the reticular endothelial system. So that means there are no hepatic impairment studies usually required or performed in for large therapeutic proteins, and there's only very limited available data available. However, the FDA colleagues published a paper a few years ago that looked at 24 therapeutic proteins where at least some data was available for patients with mild to moderate hepatic impairment, and they could show that in 19 of these 24 there was no effect at all on the systemic exposure of therapeutic proteins based on this hepatic impairment. Interestingly, however, in some individuals, there was actually an effect, and it was an effect that would be opposite to what we are used to from small molecule drugs. From small molecule drugs, if we have hepatic impairment, we often get a reduced metabolism, by that an increased systemic exposure. In this case, for therapeutic proteins, the effect that occurred, at least in a few of them, was actually the opposite. It was a reduced systemic exposure, a reduced area under the curve, and it reached, for some of them, even a value of 70%. So it was a substantial reduction. This was potentially explained by hyperglobulinemia in hepatic impairment patients. That seems to occur, and that may interfere with a mechanism, the so-called FcRn recycling, that we'll talk about in a few seconds. This has also the lack of effect on most therapeutic proteins is also further corroborated by the experience with panitumumab. Panitumumab is a monoclonal antibody that is, at least in case studies, has been given on purpose to those individuals with severe hepatic impairment with liver metastasis in their cancer that ultimately could not receive small molecule therapeutics anymore because of this renal impairment. But by administration of the standard dose of panitumumab, the exposures were comparable to individuals with normal hepatic impairment. So there's even at least the case studies where the lack of effect of hepatic impairment on the disposition and the pharmacokinetics of monoclonal antibodies is used therapeutically. One area to briefly mention is the so-called FC-mediated disposition, especially for antibodies and antibody derivatives. And this is related to the so-called neonatal FC receptor, or FcRn. And this is a little misnomer, because even though the neonatal FC receptor had first been described in neonates, it is well expressed at similar level through adulthood. So it's equally available in pediatric patients as well as in adults. Now, the FcRn receptor is a recycling mechanism that basically allows IgG molecules to circumvent the intracellular degradation process that is the fate of other therapeutic proteins. And the other therapeutic proteins are shown here in yellow, monoclonal antibodies in red. So once an endosome is formed in an endothelial cell or monocyte, for example, and is acidified, you get this pH-dependent binding of the antibody to the green molecule here, which is the FcRn receptor. And this complex can then be sorted out into a sorting endosome, recycling endosome, and ultimately brought back up to the cell surface where the pH is adjusted again to 7.4. And by that, the antibody is released out of this pH-dependent binding. So the binding affinity only is present at acidic pH. And by that, you basically shuttle these antibody molecules away from undergoing lysosomal degradation. That is the fate of the yellow other proteins that are shown here. So this is a major reason why monoclonal antibodies or IgG molecules in general have a very long circulating half-life in the body of between 21 and 24 days. This process is usually not settable as therapeutic concentrations. And the relationship is basically shown here, where you have the serum IgG concentration, so overall immune globulin G in the body, and the resulting half-life. And we are at homeostatic conditions where we have approximately 12 grams per liter in the body. We are exactly at 21 days at the half-life. So this line here is for IgG1, 2, and 4, which are also constituting all monoclonal antibodies. Now, this relationship can be overwhelmed theoretically if you give massive amounts of IgG, and this usually only occurs in one instance when you have massive intravenous Ig therapy. So if you give 20, 40, 80 grams of IgG molecules, then you start to change the overall serum concentration of IgG, and by that you would basically overwhelm this recycling process and you would get to a shorter half-life. But that is really only a very rare condition. Now, what's this different in pediatric patients? So we know that the IgG concentrations change with childhood development. They increase gradually to adult levels. And we also know that protein turnover may be different in pediatric patients. So it's been shown that lysosomal protein turnover and efficiency of FcRn recycling could be potential mechanisms for elimination of differences between children and adults after correction for size differences. Since children, including infants, are able to maintain the homeostasis of immunoglobulin, we usually assume that they can also maintain or have the same endosomal clearance pathways. And based on preliminary results, the expression of the neonatal Fc receptor FcRn is likely not substantially different between pediatric patients and adults. So that cannot be a source of differences. However, infants have substantially lower reference values for endogenous IgG subclasses, as I showed you in the previous slide, compared to older children and adults. And this, of course, may mean that you have less competition for the FcRn recycling mechanism, and by that you have a more efficient recycling, and by that a reduced clearance for endogenous IgG molecules, as well as therapeutically administered monoclonal antibodies. In addition, the protein turnover has been described to be different in young pediatric patients. It seems to be substantially higher compared to adults. For example, for low birth weight infants, protein metabolism has been described as 2 to 3 times faster than in adults when normalized for kilogram body weight. And that, of course, would mean increased clearance. So now we have several potential mechanisms. Some would result in reduced clearance, some in increased clearance of therapeutic proteins or monoclonal antibodies. So whether age effects really related to elimination cancel out or actually achieve clinically detectable differences in elimination of therapeutic proteins remains so far to be determined in future studies. So the knowledge so far is very limited and inconclusive. The last part I wanted to touch upon is immunogenicity. As you're well aware, therapeutic proteins may elicit an adaptive immune response. There may be a variety of different reasons why this occurs, sequence differences between humans and non-human proteins, different glycosylation, there may be concomitants and impurities. Oftentimes, formulation issues seem to play a role, especially the formation of aggregates, for example, in highly concentrated solutions that we have in these small volumes that we give by subcutaneous administration. There also seem to be effects with regard to dose level, certain patients with regards to their HLA type may be predisposed to have immunogenicity. So there are a variety of different factors. Now, what's the usual concern? The usual concern is that we basically form so-called anti-drug antibodies against our therapeutic protein, and these basically form immune complexes with our therapeutic protein, and those immune complexes trigger the regular endogenous elimination pathways. So that means uptake and lysosomal degradation by the reticular endothelial system, primarily in liver and spleen. And this is mediated through so-called Fc gamma receptors, primarily Fc gamma R2B2. Human platelets seem to contribute to this by expressing Fc gamma receptors and basically tagging these immune complexes for larger circulating phagocytes to ultimately take them up. So that basically means that we get an additional elimination pathway. And as shown in this little scenario here, if we have an immunogenic response, we either can have neutralizing or non-neutralizing anti-drug antibodies. The non-neutralizing do not impair the therapeutic protein to still elicit its pharmacologic effect, whereas the neutralizing usually bind at or near the binding site where the therapeutic protein binds to a target structure, and by that basically it cannot elicit the desired effect anymore. But independent of whether we have neutralizing or non-neutralizing anti-drug antibodies, they can still both form these protein ADA complexes and ultimately increase the clearance of the therapeutic protein. So that's what the major concern is. There are also other scenarios where it may decrease it, but for now I would only like to focus on the increase in clearance, which basically means reduced exposure, reduced activity, loss of efficacy. And that is, of course, an effect that's frequently seen, especially during prolonged therapy, where you have the formation of clearing anti-drug antibodies that ultimately form these immune complexes, and by that trigger then the removal of the fast removal of that therapeutic protein of interest. Shown here, this in the graphic on the right, this is one dosing interval for serum infliximab in individuals with and without detectable anti-drug antibodies. You see that those without the anti-drug antibodies can maintain the infliximab exposure nicely throughout the dosing interval, whereas those that have ADA formation, even though they have the same exposure initially, that exposure very rapidly drops down due to this formation of the immune complexes that are very rapidly removed, and then you have a prolonged period of time where you basically have essentially no infliximab exposure, which could lead to therapeutic failure. So what do we know with regard to immunogenicity in pediatric patients? So the immune system undergoes well-described developmental changes throughout childhood, as we know. And detecting immunogenicity is difficult because, of course, not every patient even has an immune reaction and forms anti-drug antibodies. So even if you look at a certain group of patients, only very few will have this immune response. And by that, differentiating whether that is now different in a adult or an older pediatric population compared to a younger one becomes very complex and challenging. So far, I think the current status is that there are a variety of monoclonal antibodies or fusion proteins for which no differences in immunogenicity between adults and pediatric populations have been described. Some of them are listed here: etanercept, infliximab, and tocilizumab. Nevertheless, there are also the exceptions that seem to have a higher ADA incidence rate in pediatric patients, adalimumab, abatacept, and daclizumab are examples for that. So this is, again, not clear whether there is an overarching principle or whether this is ultimately related to the individual therapeutic protein in question, whether there are differences between pediatric patients and adults. So many of these PK changes for therapeutic proteins in pediatrics are summarized in this graphic that we put in a review paper a couple of years ago. So we talked about the differences in absorption, the reduced lymphatic resonance time, and increased protein turnover, which may have an effect on the rate as well as the extent of absorption after subcutaneous administration. We talked about the distribution processes through convective extravasation from the vascular to the interstitial space and the drainage to the lymphatic system, where in young pediatric patients we have increased water content, increased perfusion rates, larger capillary beds, and larger capillary permeability, which may all contribute to a faster and more extensive distribution process compared to older children and adults. With regard to elimination processes, we talked about the unspecific catabolic degradation, where we have these two opposite processes, the increased protein turnover in young pediatric patients, but also the reduced competition for the FcRn recycling system due to the lower endogenous IgG reference values that we have in young pediatric patients. We do not much know about the differences in target turnover. We don't know about the ontogeny of many of the targets that our therapeutic proteins are targeting, their turnover rate, whether there are affinity differences, and whether there are differences in drug target complex internalization rate. So there's a lot of work necessary to elucidate this part. We also briefly talked about the ADA formation, but again, no clear guidance on whether there are increased or decreased ADA incidence rates in young pediatric patients. So overall, I think many approaches are nowadays used, especially to get to some of these differences between pediatric patients and adults. Physiologically based pharmacokinetic modeling has been promoted for a variety of years now, and is shown in this context here. But again, all these models are limited based on their predictions, especially in very young pediatric patients. In older pediatric patients, it works relatively well because we have a good understanding about potential differences between adults and those older populations, many of which are only driven by size differences. But if we go to very young populations, especially below 2 years of age, infants, neonates, and premature babies, oftentimes there is a large lack of knowledge in the ontogeny of some of the drug disposition processes that are relevant for therapeutic proteins. And by that, that knowledge lack ultimately translates into limited predictability for these model-based approaches. So what's my take-home message? Therapeutic proteins use different drug disposition and elimination pathways than small molecules. For many therapeutic proteins, there's only limited knowledge on which elimination pathways contribute to their disposition and to what extent. And for most of the known elimination pathways for therapeutic proteins, ontogeny information in pediatric patients is still limited but emerging. So we have some knowledge especially in the older populations, but still there is a lot of additional knowledge that's needed to further understand how developmental changes in young pediatric patients ultimately translate into these disposition processes that affect therapeutic proteins. So due to the limited understanding of the molecular and physiologic processes relevant for drug disposition and their ontogeny, approaches used in pediatric extrapolation, such as physiologically based pharmacokinetic modeling, remains challenging, with oftentimes substantial uncertainties for therapeutic proteins. And of course, my plea for more research in this arena, more mechanistic studies are needed to understand the ontogeny of disposition processes for therapeutic proteins in order to improve this situation. With that, I would like to point your attention to some book projects I've been involved with. So if you want to learn more about PKPD of therapeutic proteins, especially the pharmaceutical biotechnology textbook that is shown here from 2024 is an up-to-date reference that is available in the Springer book packages in many university libraries. So if you want to look at that, this is an electronic book. It's easily accessible and may lead to much more information if you're interested in this thematic. With that, I would like to close and thank you for your attention.
Okay. Thank you, Dr. Meibohm. We have a couple of minutes if anyone has questions. Please feel free to put it in the chat or in the Q&A box. I do have one that came up last week in our discussion of absorption modeling, relevant to mothers who may be receiving monoclonal antibodies therapeutically and then breastfeeding, and what's the risk to the baby of absorption through the gut and from the breast milk. Do you have any knowledge of that?
There are let's start with rodents. In rodents, it's very well described that monoclonal antibodies are taken up from the milk of those animals into the newborn, and that is facilitated actually also through the neonatal FC receptor. This does not seem to be a major process in humans. So whether you get a substantial uptake of antibodies from breastfeeding mothers to their children seems to be relatively limited. Where you have, of course, transfer is before birth. So the placental transfer of IgG molecules from the maternal circulation to the fetal circulation is facilitated by FcRn and is a crucial piece in establishing immunity in the fetus. Whether that occurs also through mother milk after birth, I'm not aware of at this stage. And that, of course, is well known for newborns that due to the relatively long half-life of antibodies, the IgG molecules from the maternal circulation after birth obviously persist for several weeks in the pediatric patient before they are actually getting out through their normal turnover processes.
Great, thank you. And I don't see any additional questions in the chat or Q&A. So with that, I'll thank Dr. Meibohm again for the insightful discussion on therapeutic proteins in pediatric patients. Our seminar for next week has been canceled, so we will see everyone in 2 weeks on November 19th, where Dr. Bridget Jones will be talking about developmental pharmacodynamics. Thank you very much for joining us.
Thank you. Bye-bye.